Hello everyone, I'm Vincent from GemPharmatech. Now I'm a scientist in charge of the toxicity study of transgenic mice. Today I will share about the application of genetically engineered mice in drug safety evaluation. So this webinar will be divided into four parts. The first part is the background and introduction of non-clinical safety evaluation. So in non-clinical safety evaluation, the trend of animal usage is that animal models become smaller. The usage of large animals, including non-human primates, NHP, and the canine is limited due to welfare and ethical issues. Although NHP is widely adopted in the non-clinical safety evaluation of biologics, NHP cannot fulfill all the requirements of new drugs like vaccines, gene cell therapy, and multispecific biologics in safety evaluation. Therefore, it allows small rodents, sometimes transgenic mice, be applied in the non-clinical safety evaluation. Another reason for the application of transgenic mice is laboratory animals' requirements for safety evaluation are increasing. This figure summarizes the approved IND number of innovative drugs in Chinese regulator NMPA. Increased number of IND application results in elevated demand for experimental animals, especially NHP in biologics toxicity studies. Now, the current status of experimental NHP is the first, limited availability, high cost, and ethical welfare issues. And another reason is long breeding and growth period. These factors limited the usage of NHP. And also, according to the requirements of relevant species in ISHS 6, transgenic animals expressing the human receptor should be considered when no relevant species exists. So transgenic mice could be regarded as an alternative solution in non-clinical safety evaluation of biologics. Based on the guidance of ICH files, GemPharmatech has applied transgenic mice in different safety evaluation services, including the general toxicity, reproductive toxicity, and carcinogenicity assessment. So in the second part, we will share the reproductive toxicity study case using transgenic mice. So the application of transgenic mice in the GLP reproductive toxicity mainly occurs in non-tumor biologics. The first reason is that GLP reproductive toxicity studies are often carried out in non-tumor biologics with longer administration periods and in reproductive populations. Another reason is the species advantage of transgenic mice compared with NHP. Because the breeding cycle of transgenic mice is obviously shorter than that of NHP, the time for conducting GLP reproductive toxicity using transgenic mice is 1 year, while NHP is 2 years. Moreover, NHP has limitations in carrying out EPPND studies, and follow-up of offspring to sexual maturity is usually not feasible. Even though NHP is a relevant species in some circumstances, GemPharmatech provides transgenic mice in replace of NHP to perform GLP reproductive toxicity studies. So in this case, the client would like to carry out GLP reproductive toxicity studies in human interleukin-17A monoantibody. The relevant species of this drug is cynomolgus monkeys and domestic rabbits. The customer wants to apply transgenic mice in replace of NHP. In this case, we provided DBA humanized IL-17A mice as the solution for GLP reproductive toxicity study in human IL-17A monoantibody. In order to support the technical materials that the customer replied with Chinese regulator NMPA, GemPharmatech provided detailed background data on transgenic mice. We provided gene and protein expression data to verify the expression pattern of human IL-17A in DBA IL-17A mice. The immune system validation suggested that the immune cell recomposition of DBA IL-17A mice has no significant differences compared with background mice. The reproductive status suggested that breeding status, natural breeding condition, in vitro sperm motility, and birth rate of embryo transfer of DBA-L70 mice have no significant differences compared with most widely used B6 mice. Moreover, to prove the function of human IL-17A target, we collected the pharmacological data of collagen-induced arthritis model in DBA humanized IL-17A mice, and we found that Cosentyx's commercial IL-17A antibody alleviated arthritis score compared with the vehicle group, indicating that the pharmacological activity could be exerted through humanized IL-17A target. Next, we also provided a mouse breeding program and wrote the technical support material to prove the feasibility of DBA IL-17A mice applied in GLP reproductive toxicity. Now, the customer is carrying out a reproductive toxicity assay using DBA IL-17A mice. That's all about reproductive toxicity case. Besides reproductive toxicity, the transgenic mice could also be applied in general toxicity studies. In non-GLP general toxicity study, transgenic mice could recapitulate immune-related adverse events under antibody treatment. The differences between non-clinical and clinical findings is critical in safety evaluation of immune checkpoint-related drugs like PD-1, PD-L1, or CTLA-4 targeted drugs. For example, an 82-year-old patient has obvious adverse reactions that inflammatory cell infiltration in the liver and intrahepatic duct hyperplasia after receiving ipilimumab, which is a CTLA-4 antibody. However, no obvious side effect has been found in non-clinical toxicity studies. Thus, preclinical safety evaluation of antibody drugs is in great critical. One of the reasons that result in the differences between clinical and non-clinical findings is the background chosen in animal models. Previous findings suggested that tumor-bearing biopsy mice exerted more severe organ injury in contrast with tumor-bearing B6 mice. To verify this phenomenon, we developed PD-1 CTLA-4 double humanized mouse model in both B6 background and biopsy background. Then we carried out the toxicity test with humanized PD-1 and humanized CTLA-4 antibody combination with biopsy and B6 background humanized mouse model. So we found that the survival rate of biopsy mice treated with anti-PD-1 and anti-CTLA-4 antibody was significantly reduced, while in B6 background, no significant deaths were observed. From this figure, we observed that after treatment in the antibody combination group, joint swelling, intestinal bleeding, skin inflammation, and organ inflammatory cell infiltration were observed in biopsy background mice, while in B6 background, no significant abnormality were found in growth observation. And this is the histological examination of heart in biopsy and B6 background. We found that biopsy developed more severe diffuse and more lymphocyte-infiltrated myocarditis in the heart, and also more severe lymphocyte infiltration appeared in the different organs of biopsy background humanized mice compared with B6. So in short, biopsy background humanized mice exerted antibody toxicity, which recapitulates the side effects appeared in CTLA-4 antibody treated patients. Biopsy humanized PD-1 and humanized CTLA-4 mice have been widely adopted. So the Chrome Bioscience and Charles River adopted these mice in the assessment of CTLA-4 antibody toxicity evaluation. Both of them published the poster at international conferences. So besides the application of transgenic mice in immune-related adverse effects, GemPharmatech also provides transgenic mice in GLP general toxicity studies. The first case is B6 humanized LRRC32 mice. So on 17th October 2022, an innovative anti-GARP antibody of helium completed in the GLP toxicity experiment using B6 humanized LRRC32 mice and successfully declared IND. And another case is the second generation severe immunodeficiency mouse model, NCGX. So on 1st November 2022, the HBG gene modified autologous CD34 positive hematopoietic stem cell injection of reforging medicine completed the GLP toxicity test using NCG mice of GemPharmatech and successfully declared IND. So NCGX can receive human hematopoietic stem cell transplantation without radiation for the characteristics of D, B, NK cell immunodeficiency and hematopoietic stem cell function inhibition. Therefore, it is a good model to transplant human hematopoietic stem cell study thalassemia. And in the last part, we will introduce transgenic mice in the application of carcinogenic assessment. Carcinogenicity studies are generally required for new pharmaceuticals that will be administered for 6 months or longer, or in a frequent and intermittent manner. In general, a 2-year rat study plus either a 2-year mouse study or a 6-month study in transgenic mice is required for such compounds. From 2014 to 2018, the FDA approved 213 NDA and BLA, of which 21 were conducted using RASH2 mice. Nearly 10% of marketed new drugs were evaluated for carcinogenicity using RASH2 mice. So the advantage of using transgenic mice, RASH2 mice, is including the short experiment period. It can decrease the experiment period from 2 years to half a year, and it can use these mice and reduce the influence of other factors. And in contrast, the disadvantage is it is unique in worldwide, and it is relatively expensive, and long order periods makes custom declaration and quarantine time cost. And GemPharmatech has developed transgenic mice used in carcinogenicity studies. And this mice is TG Harris mice. So TG Harris mice adopted different model strategy compared with TG RASH2 mice. The promoter of Harris in TG RAS2 mice comes from the malignant melanoma patient, while in TG Harris mice, the promoter comes from healthy individuals. So that's the main differences. And after the construction of Harris mice, we carried out the expression validation, and we found that the Harris is strongly expressed in the stomach of TG Harris mice. And we also collected the basic data, including the body weight data of TG Harris mice and the wild-type Harris mice. So we collected 65 pairs of TG Harris and 30 pairs of wild-type Harris mice from 4 weeks old to 42 weeks old. And also we have collected the blood routine and blood chemistry data. So on the basis of background data collection, we carried out the functional validation assay, collaborated with third parties. So in these cases, we found the carcinogenic sensitivity of TG Harris is similar to EA TG RASH2 mice. So from this data, we found that under MNU treatment, the TG Harris mice has the lymphoma and the skin cancer and the stomach cancers, and the carcinogenic transgenic sensitivity is similar. And we also find the tumor genesis rate of TG-Hras mice under MNU induction reaches 100%, and also the frequency of tumor genesis in the absence of drugs was much lower in Hras mice. And this is the histological data, and we can find that MNU-induced spontaneous lymphoma in spleen and thymus of TG-Hras mice, and the lymphoma also metastasized in various organs, including heart, lung, liver, prostate, pancreas, and so on. And also MNU-induced spontaneous gastric cancer in TG-Hras mice. And the last part is the survival rate analysis of TG-Hras mice. We can find that compared with G4, which is wild-type Hras mice induced by MNU, the TG-Hras mice induced by MNU has lower survival rate and are more likely to die after MNU injection. So that's all about today's webinar. Thanks for listening. Any questions and suggestions will be appreciated. Thank you. Okay, I find some questions. So the first question is, how many mice were adopted in DBA L17A reproductive toxicity studies, and which phase were carried out? So in that case, we, wait a minute. So in that case, we use about more than 1,000 DBA L17A mice in the reproductive toxicity studies, and these mice were adopted in both phase of toxicity studies. So all of the phase of reproductive toxicity studies were used. And the second question is, why do species differences between B6 and BAP6 mice appear in antibody toxicity assessment? So this is quite interesting. I suggest the underlying mechanism is the differences of MHC allies. So in BAP6 mice, it has complete MHC1 and MHC2 haplotype, while in B6 mice, the MHC haplotypes is some of the haplotypes is absent. So it will influence the efficacy. It will influence the efficiency of immune system activation. And the third part is, what is the plan for Hras mice in the future? So in the future, we will collaborate with other third parties to investigate the carcinogenicity pattern of our Hras mice and to get approved by Chinese regulator NMPA, or in future for American regulator FDA. And okay, and can data generated from Hras mice be used for IND applications? So we are making progress to use Hras mice in IND applications. We are undertaking this way, but now we have not successful cases. So any questions? Okay, I think that's all about today's webinar. Thank you all.